shifting our focus now to new tools to reduce the impact of emerging foodborne pathogens. I'm sure our next speaker doesn't need any introduction. Libby mentioned Nigel at the start because Nigel was the first director of the centre. So Nigel is the Distinguished Professor of Infectious Disease Epidemiology and Public Health at Te Whara Ora, the School of Veterinary Science at Massey, and an honorary professor in the Department of Pathology and Biomedical Sciences at the University of Otago. Nigel's currently Emeritus Director of TNIFA, the Infectious Disease Research Platform, and Emeritus Director of the New Zealand Food Safety Science and Research Centre. So, Nigel, great to have you here, and we'll let you get into your presentation. Welcome, Nigel.
Yeah, tēnā koutou katoa. It's really great to be here. I've really enjoyed the talks so far. It's been absolutely fascinating. I'll just skip to the next. Oh, yeah, of course, yeah. Yeah, so first of all, just to mention, obviously the Food Safety Centre, TNIFA, is another organisation that I'm working for at the moment. But I've got up there the ICMSF, which is the International Commission on Microbiological Specifications for Foods. And there's 23 members of that, and there's two of us in the room, actually, Roger and myself. So New Zealand's overrepresented in this international group. And what I'm mentioning that because we're writing the next book for the ICMSF, and one of the chapters in the book is on emerging risks and emerging pathogens. So it's been inspired a bit by that work. Oh dear, it's very sensitive, this. Ah, there we are. That's the end. I don't know what happened there. Certainly saved a bit more time there. You can see what I'm going to talk about now as well. Thanks. Okay, there we are. Right, there we go. Okay, so seems very sensitive. Anyway, so emerging foodborne risks. So what am I going to talk about? So pathogens are emerging all the time. So, and there are various different ways in which they emerge. So things like subtle changes in the host population, so aging populations can be more susceptible. Pathogens can pick up new traits, and I'm going to talk about a couple that have done that as well, become increasingly virulent. They can jump hosts, get into new species, and we've seen that, and I'll give an example right at the end of that as well. And also they can go into areas where they haven't been before, and certainly New Zealand's had its fair share of emerging pathogens as well. I'll try a different approach. Okay. This is maddening. So there are obviously a number of pathogens which are old and have been around for a long time, but every now and then they kind of re-emerge. And this is an example, Listeria, obviously Listeria monocytogenes has been around for a long time. This was an outbreak in Germany. We had a big outbreak in South Africa associated with polony, this sausage, and that resulted in a huge loss to the company. I think a $400 million drop in the value of that company, and also a cessation of trade in meat products from South Africa to 7 other African countries as well. So it was a huge implication for all round. Okay, what's going on? So the other thing is that about pathogens acquiring new traits. So Bacillus cereus, a well-known foodborne pathogen, has acquired some traits from a related pathogen, Bacillus anthracis, the cause of anthrax, and this has seen a change in the epidemiology. So that's an example of how evolution can affect the emergence. Okay. And then we have things like the emergence of group B streptococcus, or strep agalactiae. Another example has emerged in fresh fish samples, and was seen as an outbreak in Singapore, and very recently also now been seen in people working in the food industry as well. And this is another thing, it's not many of these pathogens are not only transmitted by the food route, but they can be transmitted by other routes as well. And then we see pathogens like the hepatitis viruses. So hepatitis A, we all know all about. Hepatitis E, causes of quite severe liver disease, has emerged around the world. And particularly associated with pigs, although there are very different strains that can be associated with other ones as well. I was just going to talk about a couple of New Zealand examples. So one of them was the emergence of a strain of Salmonella enteritidis, sequence type called ST11, which many of you might remember happened around a couple of years ago. The outbreak really started, but was detected early on. It was a disastrous desert in Auckland, and then subsequently expanded the micro database. And it turned out that it had been tracking through the poultry sector for a little while, and then it resulted in the introduction of control measures, and I think we virtually eliminated it, set on the production chain. Another salmonella enteritis, which has been around a bit longer, this is just an example with quite an unusual epidemiology. Again, here we're using whole genome sequencing in this case to show that this one, which was the cause of an outbreak in fresh produce in bean sprouts, is actually associated with hedgehogs, not just in New Zealand, but actually internationally as well, suggesting some interesting potential pathways. Another disease which you can see has emerged, or another pathogen which has emerged over time, first detected in 1993, really illustrates how emergence can appear to happen, but actually be the result of increased detection. And that's almost certainly the case with sugar toxin producing E. coli. In the early days, it was 0157. And then you can see on the right-hand side, with the introduction of culture-independent diagnostic testing, all of a sudden a whole range of different E. coli serotypes were then discovered. And this is from a recent paper by Jackie Wright. You can see with the introduction of new methods into laboratories for non-culture-based detection resulted in an increase in the number of non-0157 or untyped sugar toxin E. coli. So that was an apparent increase, which wasn't real. And this is certainly the case, though, that many of the more severe ones are still associated with two particular serotypes, 026 and 0157, but there's a large number of serotypes which now turn up routinely in laboratories and are obviously of importance for public health, from a public health point of view. So the events and processes that lead to emergence, you can see there's things like evolution, there's changes in the host or the reservoir, because all of these foodborne pathogens have some kind of animal or environmental reservoir. Amplification is another driver, so if it gets into a system and suddenly starts to multiply. Movement, if it starts to shift around the world, and I'll show an example, all of those processes can then lead to the emergence of a new or emerging or re-emerging foodborne pathogen. So one example I'm just going to use and illustrate with that diagram is Vibrio parahemolyticus. There was a sequence type that was detected, caused a major outbreak in China. We have other sequence types also which have emerged in a similar way. Highly pathogenic strains. And what most likely happened, you might have had evolution occurring in those reservoirs, host changes, great range of hosts in terms of the shellfish that were infected, changes in the harvest season, those are host changes, and then the impact of climate change, changes in sea level and salinity then resulted in movement, and that resulted in amplification and emergence. So not every pathogen behaves in the same way, but you've got that kind of patterns that can lead to emergence. So what are the new tools? There's a whole range of new tools which are coming on board, which help us with dealing with emerging and existing pathogens as well. And I'm just going to focus on a couple of them, and some have already been mentioned, so I can talk through very quickly. But genomics and meta-omics technologies. So we're talking about whole genome sequencing, which is not new anymore. It's actually routinely adopted in many parts of the food industry, and certainly within public health as well. Meta-omics, I'm talking about metagenomics, metatranscriptomics, metaproteomics, and metabolomics as well. So there's all these 4 different ones. I only talk about 2 of them, and that's metagenomics and metatranscriptomics, because these are the ones that seem to be emerging at the moment. Many different applications, from diagnostics, so detecting existing and emerging pathogens, source attribution, where does this foodborne infection come from, antimicrobial resistance profiling, and also looking at transmission within processing plants as well. We reviewed that for a publication there, which you might be interested in. And this is just setting out a framework for using, in this case, whole genome sequencing within a system from the animal reservoirs all the way through to human infection. So it's a kind of schematic showing how these technologies can be implemented within the system as a whole. And we know from a public health perspective, the impact of whole genome sequencing has resulted in the detection of more smaller outbreaks. So what was traditionally, it was in the lower resolution sequencing would have been a single large outbreak. We now know are multiple smaller outbreaks, but the impact of knowing that means that you can intervene earlier and reduce the duration and size of each of those individual outbreaks. So this is one of the values of whole genome sequencing. And we've applied these tools and techniques for the Salmonella outbreak, which I mentioned earlier. This is a very recent publication. Non-culture based detection. So this is where things like metagenomics I was talking about earlier, and I'll talk about some other methods as well. This is a really great review by Craig, Joe, and Lucy as well from PHF. Highly cited now and really is a very valuable resource for anyone interested. So metagenomics is not culture-based. It's basically just extracting all the DNA or the RNA, the nucleic acids from a sample, and then using a range of different tools to then characterize what's in that sample as a whole. And it's a way of looking at the entire microbiome of, it could be a food product like cheese or yogurt or something like that, or it could be a whole processing environment. And basically what you're wanting to do is look at what's called the dysbiosis, or a change in that microbiome, which might indicate a range of different drivers, which could signify or identify the emergence of a new problem or a new pathogen. And those are just some quotes from that paper. And Mars and IBM joined together to do this project, which I haven't actually seen any outputs from this project yet, even though it was set up a while ago. So I suspect it hasn't worked. But nevertheless, it's being taken seriously by other organizations. So EFSA have done this whole analysis of ways in which standardization, harmonization of looking at these entire food chain microbiomes might be done. And if you're interested in that, this is a very recent publication. Metagenomics, again, early stages, and I think it's still yet to see any potential, but nevertheless, it's something you might see come on board. So looking at changes in those microbial populations, it's culture independent. And there are some examples in this publication that you can look at. Routine monitoring, for example, of raw ingredients coming into a plant, so you can use that to determine what might be the source of contamination in a final product. And also, one of the other things that we're doing currently at Massey is extracting whole genomes. You can actually extract whole genome sequences from metagenomic data as well. Metatranscriptomics is looking at RNA. So this is looking at genes that are transcribed. So this has the advantage as you can also use it to try and detect live organisms. The disadvantage of metagenomics is it will pick up both live and dead organisms. But there are many hurdles to this technology. So it'll be interesting to see how this develops. So one of them is if you're detecting an entire microbial complement of a sample, it can add to the portfolio of things that you need to worry about. You can see that. And this is recognized as a huge potential issue. And obviously storing the data can, and that's the last thing that you want from a food industry perspective, is to increase your work. Costs can increase, particularly with the downstream bioinformatics. There's a lack of standardization and harmonization, although there's work coming on board to look at that. And with metagenomics, you're not necessarily looking at live organisms. So there's a number of disadvantages as well. That was metagenomics. Metatranscriptomics, there are other techniques. So this is something that's being used in wastewater epidemiology at the moment. This is hyperplex PCR. So these are arrays with over, well, around about 100 different targets on them. So you can look at many, many different things. So this is one example, a company that's selling this particular product. You may remember during COVID, we all did rats, and we could still do those rats as well. So these lateral flow assays are also becoming more cheaper and more sensitive and more specific as well. So we're moving now into the top right-hand corner with the next generation of lateral flow assays. And I think some of these will be, if they're not already being used, will be used more in the food industry as well as for public health purposes. Isothermal amplification. So this is again something, active areas of research using these techniques like LAMP and RPA. You don't need a thermocycler PCR to do this. It can be done at much, much lower temperatures. RPA temperatures of around 30 to 40 degrees. So this is ways of amplifying DNA or RNA, which it then can be used for further detection purposes, particularly if it's combined with things like the CRISPR-Cas assay. So we're developing assays at the moment that combine RPA with CRISPR-Cas to try and develop highly sensitive and specific and rapid approaches. Only to find that it had already been done before. So this is an example of an assay which was published very recently, much to the disappointment of the student who's working on this project. But nevertheless, it illustrates the potential for a very rapid assay. So in this case, less than an hour, combining the RPA with the CRISPR-Cas into a device that can be used in this case to detect not only Clostridium perfringens, but also toxin type that as well. AI has been talked about extensively, so I won't go into great detail, but just to mention, obviously has multiple applications. I do think there's been a bit of hype, particularly around the use of large language models. They're still giant lookup tables at this stage. They're not really thinking in the same way that we think. But other applications are showing huge promise. But fundamentally, it's methods and software that enable machines to perceive their environment, learn from it, and take intelligent actions to help you achieve defined goals. So that's basically all it does. It's a huge advance, but that's what it does. There's many ways, many sources of data that you can put into these models. And obviously one of the issues is sensitive data you don't want to be uploading to international databases, but it's costly to buy your own system. And I'd be interested to know how in particular industries using these kinds of tools at the moment. Examples in food safety, we've been working on machine learning models for source attribution and applied those in a number of different settings, but also things like hyperspectral imaging for food safety purposes, identifying restaurants violating health codes, so using online data to be able to do that. And the Chinese are certainly using it for trying to predict food import firms that are likely to fail site identifications as well. So there's various ranges of which it can be used. But just to point out, if you're really interested in this, it was a great talk by Jared earlier on about the applications of AI. But this is specifically AI for food safety. And this publication has just come out from the FAO. And it's got quite a lot of it. It's not a long publication. It's got a lot of useful information. And lastly, we know for AI purposes that combining it with robotics, this particular robot dog, the dog called Spot, being deployed in a number of different organizations to detect it. It can get auditory, visual, infrared, thermal imaging, and it can be used for food safety purposes, but also for health and safety purposes as well, and is being used. Now, lastly, I just wanted to talk very briefly about an emerging issue, which I think is probably on everybody's minds at the moment, but just this is part of my other work with Tanifa, is around pandemic planning and preparedness. And pandemic planning, one of the really big issues that we're thinking about at the moment is the emergence of avian influenza. And obviously with pandemic planning, communicable disease, foodborne illness, potential foodborne pathogens that might be pandemics is just one biological hazard within the 28 hazards of being overseen and looked after by the DPMC at the moment. So this is just one piece of work within a much larger, within the national risk framework. Just in the last year, we're beginning to see avian influenza move around the subantarctic region. Oceania still is the only continent without the emerging strain, the one that's circulating around the world. It's called 2344B strain, and it's one that's causing a lot of concern at the moment. And one of the reasons is in North America, we've seen outbreaks in cattle farms. So in dairy herds in particular, over 1,000 dairy herds now have seen the crossover, and it's happened more than once, from wild birds and poultry into the cattle system. And that's resulted in a major effort to try and deal with it. And because it also affects people, it's not a severe illness at the moment, although I think one person has died from this strain. You're seeing it in workers, in particular, it has highly concentrated in milk, in the udder, and probably just through splashing in the milking parlour is what's causing some of these symptoms and signs in workers. And it's probably hugely underreported. And one of the reasons may well be not wanting to identify your farm as being infected, but also many of the workers are migrant workers as well. So there's a lack of willingness to report illness. Situation here, we did have bird flu in a farm at the end of last year, but that wasn't this circulating strain. That was one that we grew locally. So it was a different situation. But there is a huge effort going in to try and protect, particularly our Tonga wildlife species, so vaccinating some of our captive breeded birds. But there's a real issue, elephant seals at the moment, other sea lions dying. 96% of the population of elephant seals have been wiped out in many colonies. Imagine if that got into New Zealand, into the sea lion population. So it's an example of a multifaceted issue. It's not a food safety issue, so I think we need to bear that in mind, but it's something of concern, and it will be raised as an issue by the general public too. It's likely to come in through either wild birds or wild mammals. It's likely to get into the poultry sector and potentially into cattle as well if it crosses over. So we need to be prepared. And there are many, many aspects to preparing for a pandemic zoonosis of that kind, and taking it very seriously at this stage. And so just to summarize, and apologies again for the issues with my slides at the beginning there, but foodborne pathogens are emerging and re-emerging continuously. We're seeing them popping up, new ones, old ones, re-emerging. There are many, many drivers, including climate change, which we talked about earlier as well. Detection, leadership, all of these things, surveillance are really important for this, for dealing with emerging pathogens. Open sharing of data, so talking about metagenomic data, and the use of metagenomics is of interest. We can see on that diagram on the right-hand side that metagenomics is now, we're seeing development of standardization and harmonization methods for that, not so much with some of the other omics technologies, but there are real downsides to this technology as well, increasing costs, increasing the number of pathogens to consider, issues related to privacy, stewardship, ownership, and security. All of these could potentially raise threats and liability for food companies as well. So it's an emerging technology that needs to be watched very carefully. And similarly, issues with the use of artificial intelligence. And the last thing was about avian influenza, an existential threat that we need to take seriously. And I think it will arrive at some stage in the next year or so. I'm sorry to say.